Hello, students. This lecture will focus on how microorganisms communicate with each other. The lecture material can be found in Chapter 22 of your textbook. In this lecture, we will learn the different diffusible signaling molecules in bacteria, in specific signaling systems such as competence and endosporulation in Bacillus subtilis, virulence genes in Staphylococcus aureus, and bactericin production by Lactococcus lactis. How do cells communicate with each other? Cells communicate with each other by sending and receiving signals. The communication process involves a sender cell, also called the secreting cell, and the recipient cell or target cell. The secreting cell talks to other cells by synthesizing and producing chemical signals. The target cell, on the other end, listens by responding to the chemical signal. This process is commonly known as cell-cell communication or cell-to-cell signaling. Cell signaling is part of a complex system of communication among cells that governs basic cellular activities and coordinates cell actions. Cells typically communicate using chemical signals. These chemical signals, which are proteins or other molecules produced by a sending cell, are often secreted from the cell and released into the extracellular space. There, they float like messages in a bottle over to neighboring cells. The chemical signal is received by binding to specific receptors located in the cell membrane of the recipient cell or target cell. The chemical signal then enters the cell membrane, causing a signaling cascade that alters gene expression or cellular behavior. It is very important to realize that for signal transmission to occur, each signaling molecule must have a specific receptor on responding to the cell. Not all cells can hear a particular chemical message. In order to detect a signal, that is, to be a target cell, a neighbor cell must have the right receptor for that signal. When a signaling molecule binds to its receptor, it alters the shape or activity of the receptor, triggering a change inside of the cell. Signaling molecules are often called ligands, a general term for molecules that bind specifically to other molecules, such as receptors. The message carried by a ligand or signaling molecule is often relayed through a chain of chemical messengers inside the cell. Ultimately, it leads to a change in the cell, such as alteration in the activity of a gene, or even the induction of a whole process, such as cell division. Thus, the original intercellular or between cells signal is converted into an intracellular or within cell signal that triggers a response. Signaling molecules produced by bacteria encompass a wide range of chemical structures. Signaling molecules exist as oligopeptide pheromones or acyl homoserine lactone, cited in this table as AHL. In general, gram-positive bacteria such as Bacillus subtilis, Enterococcus faecalis, lactic acid bacteria, Staphylococcus aureus, and Streptococcus pneumonia utilize oligopeptide pheromones as signaling molecules, while gram-negative bacteria such as Vibrio, Agrobacterium, and Pseudomonas species use acyl homoserine lactones. Signaling molecules are mediators of gene expression and regulate different cellular activities such as competence, DNA exchange, bioluminescence, and pathogenesis. Oligopeptide pheromones are small peptides ranging from 5 to 50 amino acids. These pheromones are processed from larger peptides within the cell. They serve as signaling molecules among gram-positive bacteria. For instance, Lactococcus lactis, a gram-positive bacterium, utilizes nisin A, a signaling molecule, for bacteriocin production. Bacillus subtilis, another gram-positive bacterium, utilizes COMX and CSF as signaling molecules to trigger DNA uptake and sporulation. Oligopeptide pheromones must bind to receptors to initiate a signal cascade. There are two types of receptors, the cell membrane receptor and the cytoplasmic membrane receptor. The cell membrane receptor is located in the cell membrane or inner membrane of the cell. Cell membrane receptors can be membrane transporters or oligonucleotide permease or histidine kinase. Cytoplasmic membrane receptor is a receptor located in the cytosol. Cytoplasmic membrane receptors can be regulatory proteins, activator proteins, or secondary messengers that regulate the transcription of specific genes and may be activated or inactivated by pheromone binding. Oligopeptide pheromones first bind to the cell membrane receptors of the recipient cell. The binding of the pheromone to the cell membrane receptor generates a transmembrane signal that initiates a signal cascade within the cell. The oligopeptide pheromone then enters the cell and then binds to the cytoplasmic receptor, which may be activated or inactivated by pheromone binding. Ultimately, this leads to a change in the cell behavior. The development of competence in sporulation of a gram-positive bacterium Bacillus subtilis is mediated by oligopeptide pheromones. It involves the oligopeptide pheromones COMX and CSF. COMX is a 10 amino acid oligopeptide pheromone. CSF is a 5 amino acid oligopeptide pheromone. Competence is the ability of a cell to take up extracellular or naked DNA from its environment. In order for Bacillus subtilis to take up DNA from its environment, its competence must be induced. COMX is involved in this process. Sporulation is the ability to produce spores. CSF is involved in this process. Bacillus subtilis is a spore-forming rod-shaped gram-positive bacterium. When nutrients are depleted, it produces and matures into a spore. When nutrients become available, it germinates into a vegetative cell. This microorganism is commonly found in soil and has been used as a model organism for competence in sporulation. Bacillus subtilis is among naturally competent microorganisms. Natural competence is the ability to bind and transport exogenous DNA into the recipient cell. Natural competence is one way in which genes can be transferred between closely related bacteria. Competence in sporulation usually arises at specific growth stage of a culture. As you can see from this growth curve, competence and sporulation occur during the late log phase or late exponential phase. Competence and sporulation are alternative developmental phase in the cell. They both tend to occur at the end of exponential phase when the cell densities are high. They are regulated by oligopeptide pheromones secreted by the cells into the extracellular medium. At low cell densities, very little oligopeptide pheromones are secreted, therefore, there is no transcription of competence or sporulation genes. At high densities, high concentrations of oligopeptide pheromones are secreted, which trigger transcription of competence or sporulation genes. High concentrations of pheromones are necessary for competence in sporulation, and this occurs during late exponential phase when cell densities are the highest. Bacillus subtilis, like many organisms, has mechanisms for sensing and responding to high cell density. In Bacillus subtilis, both the initiation of sporulation and competence development are regulated in part by high cell density. There are two peptide pheromones that accumulate in the extracellular media, high cell densities, or during the late exponential phase of growth. These pheromones are the COMX and CSF, which stands for competence stimulating factor. COMX is a 10 amino acid pheromone. It is involved in the induction of competence, and its response requires a two-component regulatory system, which requires a sensor kinase and a response regulator. COMP is the histidine kinase located in the cell membrane. COM-A is a cytoplasmic response regulator or transcription factor. The cellular response to COMX pheromone requires the membrane-bound histidine protein kinase, COMP. In the presence of COMX pheromones, COMP autophosphorylates and donates phosphate to the response regulator or the transcription factor, COM-A. When COM-A is phosphorylated to COM-AP, it stimulates transcription of genes in the competence pathway. CSF, the other oligonucleotide pheromone, also accumulates in the medium as Bacillus subtilis grow to a high density. CSF is a 5 amino acid pheromone that is involved in competence and sporulation. The cellular response to CSF requires transfer back into the cell via oligopeptide permease. CSF stimulates accumulation of COM-AP or phosphorylated COM-A and competence gene expression by inhibiting activity of RAPC, a phosphatase that dephosphorylates COM-AP. Both COMX and CSF increase the amount of COM-AP or phosphorylated COM-A, which leads to the expression of competence genes. COMX stimulates competence gene expression by affecting the activity of the histidine kinase COMP, while CSF stimulates competence gene expression by an opposing phosphatase RAPC. So how is competence regulated by oligopeptide pheromones in Bacillus subtilis? The oligopeptide pheromone COMX binds to histidine kinase COMP, causing it to autophosphorylate and initiate phosphorylase. COM-A, the cytoplasmic response regulator, receives the message from COMP and becomes phosphorylated to COM-AP. The phosphorylation of COM-A to COM-AP leads to the transcription of genes in the competence pathway. CSF, the other oligonucleotide pheromone, also accumulates in the medium as Bacillus subtilis grow to a high density. CSF enters the cell via an oligonucleotide permease and inhibits RAPC, a phosphatase that dephosphorylates COM-AP. Thus, both COMX and CSF increase the amount of COM-AP or phosphorylated COM-A, which stimulates expression of competence genes. CSF is involved in competence as it increases the amount of COM-AP or phosphorylated COM-A, which ultimately stimulates the expression of competence genes. CSF also stimulates sporulation. CSF is the extracellular peptide pheromone that stimulates sporulation. At low concentrations of 1 to 5 nanomole, CSF stimulates the competence pathway and inhibits sporulation. At low concentrations, it prevents dephosphorylation of COM-AP, allowing the expression of competence genes. At high concentrations, greater than 20 nanomole, it inhibits competence genes and stimulates sporulation genes. At high concentrations, it prevents the phosphorylation of the histidine kinase COMP, inhibiting the phosphorylase and the ultimate expression of competence genes to occur. Sporulation in Bacillus subtilis is controlled by a complex gene regulatory circuit that is activated upon nutrient deprivation. The system includes two sensor kinases, KIN-A and KIN-B. KIN-B is a sensor kinase located in the cell membrane. It senses low nutrient concentration. Kin A is a sensor kinase located in the cytosol. It senses intracellular CSF concentration or CSF concentrations in the cytosol. The system also includes two additional phosphotransferases, SPOOF and SPOOB. These two phosphotransphorases activate the master transcription factor or response regulator SPOOA. SPOOA is the primary positive transcriptional regulator for sporulation genes. The key to understanding the initiation of sporulation is understanding the mechanisms of the signal transduction pathway. One striking feature of the signal transduction pathway is a complex phosphorylase to activate SPOOA. The phosphorylase is a complex variation of a two-component regulatory system. It includes phosphotransferases that transfer the phosphoryl group from the sensor kinases to the ultimate target. When a concentration of CSF in the extracellular environment is high, which occurs when nutrients are depleted, Kin B, a membrane-associated histidine kinase, and Kin A cytoplasmic histidine kinase, autophosphorylate. The phosphoryl group comes from the ATP donor, which is hydrolyzed to ADP, converting Kin B to Kin BP and Kin A to Kin AP. The phosphoryl groups from Kin B are then transferred from SPOOF to SPOOB and in turn are transferred to the transcriptional activator SPOOA. SPOOAP, or the phosphorylated SPOOA, stimulates the transcription of sporulation genes. In addition to the histidine kinases Kin A and Kin B, three phosphatases regulate the levels of SPOOAP, or phosphorylated SPOOA. They are the ROP-A, ROP-B, and SPOOE phosphatases. ROP-A dephosphorylates SPOOFP. SPOOE dephosphorylates SPOOAP. ROP-B prevents SPOOFP dephosphorylation. When cell density is high, the secreted CSF pheromones return back into the cell and inhibit the ROP-B protein, which is a phosphatase that prevents SPOOFP dephosphorylation. This allows the phosphorylase cascade to happen, which ultimately leads to the expression of the sporulation genes. When nutrients are plentiful and the CSF concentration in the cell is low, ROP-A and SPOOE dephosphorylates SPOOFP and SPOOAP respectively. This result prevents the phosphorylase cascade and the eventual expression of sporulation pathway. Moving on to Staphylococcus aureus. Staphylococcus aureus is a gram-positive spherical-shaped bacterium. The bacterium produces many virulence factors that contribute to pathogenesis. These virulence factors include enterotoxin B, which is an enterotoxin commonly associated with foodborne illness, hemolysin, a toxin that lyses red blood cells, and leukocidin, a toxin that kills white blood cells. Staphylococcus aureus utilizes oligopeptide pheromone to regulate pathogenesis. The oligopeptide signaling molecule it uses is called AGRD or AIP, which is an 8 amino acid peptide. One of the toxins the Staphylococcus aureus produces is the staphylococcal alpha toxin. Staphylococcal alpha toxin induces apoptosis in certain human immune cells, such as lymphocytes, leukocytes, and monocytes. The figure shows you its host cell membrane. As you can see from this figure, it forms a pore or a hole in the cytoplasmic membrane of its host, releasing the contents of the cell and allowing the influx of extracellular material and efflux of intracellular material. The infected host cells eventually swell and lyse. The expression of many virulence genes in Staphylococcus aureus is dependent upon the AGR regulatory system. AGR stands for accessory gene regulation, which responds to an oligopeptide type quorum sensing signal. The regulatory system includes AGRC, which is the histidine kinase, AGRA is the response regulator, and AIP is the extracellular oligopeptide pheromone encoded by AGRB gene. The expression of AGRB gene leads to the synthesis of AGRB protein, also known as the oligopeptide pheromone AIP. The expression of AGRA gene leads to the synthesis of AGRA protein, also known as the response regulator. The expression of AGRC gene leads to the synthesis of AGRC protein, also known as the sensor kinase. At high cell density, Staphylococcus aureus cells produce the signaling molecule AIP at high concentrations. The AIP binds to the sensor kinase AGRC and stimulates autophosphorylation. The phosphoryl group from AGRC is passed to the cytoplasmic response regulator AGRA and becomes phosphorylated. AGRAP, or the phosphorylated AGRA, does not appear to bind with DNA, but it stimulates expression of RNA3 genes by interacting with a DNA-binding transcription factor. AGRAP leads to the expression of genes involved in pathogenesis, such as production of toxins and adhesion proteins. At low cell density, very little AIP is synthesized. This result prevents the expression of genes involved in virulence. Moving on to Lactobacillus lactis. Lactobacillus lactis is a gram-positive spherical-shaped bacterium. It produces a bacteriocin called nisin that inhibits the growth of many gram-positive bacteria. Bacteriocins are antimicrobial peptides that help bacteria fight competing bacteria in microecological systems. Nisin is an example of a bacteriocin. It is considered to be class 1 bacteriocin. Nisin is a natural antimicrobial agent with activity against a wide variety of gram-positive bacteria, including foodborne pathogens such as Listeria, Staphylococcus, and Clostridium. It is widely used in food industry as a safe and natural preservative. Class 1 bacteriocins, such as nisin, have a dual mechanism in killing their target cells through inhibition of cell wall synthesis and pore formation. Both mechanisms are facilitated by the binding of the nisin molecule to the lipid tube, which is the main transporter of peptidoglycan subunits, the building blocks of the bacterial cell wall. At lower concentrations, nisin prevents the proper cell wall synthesis, which leads to cell death. At higher concentrations, nisin binds the lipid tube that initiates membrane insertion and pore formation, which leads to the leakage of cellular components such as ions and ATP. The production of nisin by Lactococcus lactis is stimulated by extracellular oligopeptide pheromone nisin-A. Nisin-A is a 34 amino acid peptide. The regulation of nisin biosynthesis is controlled by a two-component regulatory system composed of a histidine kinase, Nis-K, and a response regulator, Nis-R, where the nisin-A molecule itself serves as the peptide pheromone. The Nis-K protein senses the nisin-A molecule, causing it to autophosphorylate and subsequently transfer the phosphoryl group to Nis-R. The phosphorylated NIS-R then initiates the transcription of the NIS-A gene cluster by activating the promoters. Acyl homoserine lactones are a class of signaling molecules involved in bacterial quorum sensing. They were first discovered as signaling molecules in several marine Vibrio species and are produced by other gram-negative bacteria such as Agrobacterium tumefaciens and Pseudomonas aeruginosa. They regulate cellular processes such as bioluminescence, virulence, DNA exchange, pathogenesis, and biofilm formation. DNA exchange in Agrobacterium tumefaciens utilizes acyl homoserine lactones as signaling molecule. Agrobacterium tumefaciens is a gram-negative rod-shaped bacterium. It is closely related to symbiotic nitrogen-fixing bacteria, which dwell at root nodules in legumes. Unlike most other soil-dwelling bacteria, it infects the roots of plants to cause crown gall disease or tumor formation in over 140 plant species, including oak trees, tomato, and tobacco plants. Agrobacterium tumefaciens is a soil-borne plant pathogen. It contains TI plasmid, or tumor-inducing plasmid, which is responsible for its virulence. The TI plasmid contains several genes, including genes that mobilize DNA for transfer to the plant and virulence genes. It also contains tDNA, or transfer DNA, which carries the gene for tumor formation. The tDNA has two genes, the ONC gene and the opine gene. The ONC gene is responsible for the tumorigenesis. The opine gene is responsible for the synthesis of modified amino acids called opines. The tDNA from the TI plasmid of Agrobacterium cells are transferred to the host cell via conjugation process. The conjugation process requires the expression of the TRA genes or transfer genes. The expression of TRA genes is governed by the TRA-R, TRA-I quorum sensing system. The TRA-R, TRA-I system consists of TRA-R gene, which encodes the activator protein that stimulates the expression of genes required for conjugation, and the TRA-I gene, which encodes the production of the signaling molecule acyl homoserine lactone. When the density of Agrobacterium tumefaciens is sufficiently high, the transfer of the tDNA is stimulated by bacterial quorum sensing called the TRA-R, TRA-I quorum sensing. TRA-I synthesizes 3-oxo-C8HCl signaling molecule. The signaling molecules diffuse out of the cell. When high concentration of signaling molecules is reached, they diffuse back into the cell. The signaling molecule bind and activate TRA-R and triggers transcription of TRA genes.